There are a small group of pioneers who have explored a world too small to see. What they found there has answered some eternal questions. What is life? How is it made? Why are children like their parents? Their work has triggered a revolution that may make the iron and silicon revolutions seem like footnotes in human history. This series is their story, the story of the people who unlocked the secret of life. This is DNA, what it actually looks like according to the latest scientific data. It is without doubt the most powerful substance on Earth. The order of its chemicals determines whether a child has her mother's eyes or her father's smile. Until 50 years ago, almost no one knew of its existence, but that all changed when two unknown scientists ran into a pub in Cambridge shouting that they'd found the secret of life: Jim Watson and Francis Crick. If you go to Cambridge and walk around the grand old buildings, you'll hear of the achievements of Francis Bacon, Alfred Lord Tennyson, and Isaac Newton. But if you ask about Watson and Crick, you'll hear rumors of improper behavior, skullduggery, even plagiarism. But what if you go to the people themselves? What do they have to say? Francis Crick lives in a house on the edge of the Mojave Desert in California, and he's become a little reclusive when it comes to discussing DNA. Some say it's because he's ill. Others say he finds the whole subject uncomfortable. Or maybe he's just busy with his more recent work, studying the chemical nature of dreams. Whatever the reason, he no longer gives interviews. But Jim Watson does. Today, he's a 74-year-old, world-famous multimillionaire. But he was just a 25-year-old junior researcher when he explained life in scientific terms. Still, all these years later, one gets a sense of the brash young man who was planning to overthrow the old ways of thinking.
If you looked at Cambridge, we were products of God. And Darwin wasn't celebrated as the most important person. The statement that life could be understood finally in terms of molecules, people would say it was a hypothesis. Until you could explain heredity in terms of molecules, someone could always say there was something else. So Francis and I, what united us is, we just thought religion was wrong. Not necessarily silly, but wrong. There was no God, and humans had to make our own rules, not just obey rules because someone said they came from God. You realize that that people thought we were slightly crazy, but we didn't think we were. So we thought the other people were dull.
Dull or not, the other people at Cambridge didn't hold up much hope of Watson and Crick doing anything at all, let alone finding the secret of life. Watson, the awkward American kid, and Crick, a splendid talker who had never quite managed to finish his PhD. They were seen as lazy jokers. To appreciate what they did, we have to imagine ourselves in the 1950s, when all that was known about life was what could be seen down a microscope. Cells dividing. they could somehow magically divide to form plants or panthers or people. But how? There had to be some kind of script or instructions telling the cells what to do. And it had to be here, in the chromosomes. It was known that chromosomes were made of two discrete ingredients called proteins and DNA. Most people expected to find the answer in the proteins. Proteins are very complex and made up of many different chemicals. So they distracted the best minds. But Watson and Crick decided to look at the simpler DNA. DNA is composed of only 4 ingredients. They thought if they could work out how the atoms of these 4 ingredients were arranged in physical space, they might be able to work out what they did. They had a hunch that the 3-dimensional structure might reveal its function. But while Watson and Crick had never found the structure of anything, 60 miles away in London, there were another pair of scientists who had. Rosalind Franklin has been called the Dark Lady of DNA. She died without ever getting the credit for her part in the discovery. Some say she was betrayed by her colleague at King's College London. If you go to King's, along a remote corridor and down these stairs, 50 years on, you'll still find her colleague, Maurice Wilkins. And on the walls of his office are clues which hint at what he's been through. During the Second World War, he helped to create the atom bomb. The night it was dropped on Hiroshima, he was at a party celebrating the culmination of his work when a man came up to him and said something that would change his life.
It was being Monday when the bomb went off, and he said, I call it Black Monday. I always hoped it wouldn't work. I sort of stood there and felt a bit small, and then said, yes, I think you're quite right. But it did work, and so we are living kind of in the aftermath of that.
Disillusioned with the science of death, he chose the science of life instead. And that's why he chose to look for the structure of DNA.
Well, here is one of the X-ray generators we're using in this work. Here is the X-ray tube.
Wilkins' strange contraption is in fact a kind of camera. It's a technique called X-ray crystallography. A crystalline form of DNA is placed inside the camera, and when X-rays are fired through it, they scatter onto photographic paper and form a regular pattern. It's a bit like shining a spotlight at a chandelier. Light hits the crystals and then diffracts onto the wall. Now imagine you can't see the chandelier, and you can just see the light on the wall. From that, you have to guess the shape of the chandelier. And that's what these photographs are. Taken by Maurice Wilkins, they were the first clue to the structure of DNA. But his boss realized he was onto something big and decided to bring in an expert, Rosalind Franklin. And suddenly, it wasn't just Wilkins taking photographs of DNA. So, Watson and Crick were in Cambridge, Wilkins and Franklin were in London. But there was someone else lurking out there, someone with a formidable reputation, an American called Linus Pauling. He died years ago, but his son was involved too, and we found him in the middle of Wales, miles from the nearest town. In this house is Peter Pauling.
It's very often in science that the times are right and different people have the same idea, at roughly the same time. And getting in first somehow is important.
Although they didn't all realize it, these 5 characters were in a race. Peter Pauling knew them all and watched this piece of history unfold. His father had the best credentials, a double Nobel Prize winner.
Pa did very few things by accident. He did things, he had a reason for doing things. To Pa, DNA was just a substance like sodium chloride.
Linus Pauling looked for the structures of many molecules, and he usually found them too. That's why in the world of 1950s science, he was one of the most recognizable figures.
I like to understand the world. I like to learn about new ideas. But I also like very much having new ideas myself or making discoveries myself. This pleases me immensely.
He had a different approach. He was going to guess the structure by building ball and spoke models. The balls represent the atoms. The spokes are how far apart they must be from each other, according to the laws of physics. So Linus Pauling would then try to see how they fitted together. Solving a structure this way was like doing a 3-dimensional jigsaw puzzle. So Watson and Crick had a choice, to do the painstaking X-ray work or try their luck building models like Linus Pauling. For them, the choice was simple.
To build models. It was only a question when you started to build the models. Would you do it after you collected years of experimental data, or would you try and build the model with a minimum of data?
So they went for model building with the minimum of data. Some might say a more leisurely approach, but that was the Cambridge style. And Watson and Crick seemed to be the epitome of that, whiling away the hours, chatting about the secret of life.
People would laugh at us and say, oh, we weren't doing experiments. Just take these long walks at lunch and constantly talking instead of experimenting.
Happy days.
People see you do no experiments. It was sort of thought, you know, we were parasites. Other people did the work and we got the glory. But the truth was, you know, complicated.
In fact, Watson and Crick were asking all the right questions.
What was common to all forms of life? And what finally seemed to be common to all forms of life was there was a script. We had been thinking it was DNA, but we didn't know the shape of the script, and always the big problem was the copying. Who copied it? There weren't any, you know, little monks inside the cell copying the script.
By not getting bogged down in the details of experiments, their minds were free to concentrate on the big ideas. Their constant talking was in fact a way for them to distill their theories. 3 teams, 3 different approaches, and Watson and Crick weren't the favourites.
I would say that Watson and Crick were number 2. And at that time, I would put my money on this crowd.
King's College London should have been the ideal place to study DNA, but trouble was brewing. While Maurice Wilkins blended into the shadows, Rosalind Franklin was making a big impression on those around her.
She was of medium height and had black hair, which she wore straight, in no particular arrangement. But she had the most startling dark eyes, which showed the intense nature of her personality. Most of the young men who worked with her were half in love with her.
One of these young men was Raymond Gosling. At the time, he was a lowly lab assistant working for Rosalind Franklin.
Looking back on it, I think I was very privileged to have been there. I only wish I had known at the time that it was that important. I might have remembered more and worked harder, and who knows, I might have tried building a few models secretly. Model building was in the air, but her view was you could build models all day, but how did you prove which one was right? On the other hand, if you made the measurements, you did all the corrective geometry, and you put them into the equations, you would let the data speak for itself. And out of that would come a definitive structure.
But there was a problem. Wilkins was under the impression that DNA was his project, but their boss had told Franklin it was hers.
No mention was ever made of the fact that Wilkins was the overarching person concerned in the lab. She certainly felt she was coming in, she was taking over that diffraction work.
So, at opposite ends of the corridor, Maurice Wilkins and Rosalind Franklin worked on DNA. Occasionally, they would announce their results to the department. One afternoon in November 1951, Franklin was to reveal her latest DNA data to a select group of King's College scientists, and one outsider.
Well, this is where I came in. Early November 1951, to hear Rosalind Franklin talk about her newest results on DNA. And I was terribly keen to know what she'd done because I wanted to build a model, and I thought I would learn possibly something about the structure. There was probably 30 people in the room, and I slipped in, so it was inside us, like a spy. And Rosalind, was seemingly much in control. I generally never took notes. My memory was good.
Having taken in as much as he could of the X-ray data, Watson rushed back to Cambridge to tell Crick what he'd heard. For the next 2 weeks they worked on a model, and on the 28th of November 1951, Watson and Crick announced that they had found the structure of DNA.
Francis rang me up and said, "We made a model, come have a look." So I went to the others and collected them, and we all went up.
It was a pretty ugly structure. Francis liked it. I don't know. He called up the people at King's and said we'd done something clever, and I was a bit worried.
Feeling apprehensive, the King's team left for Cambridge. Rosalind Franklin took one look at the model and
She laughed at them, much to their discomfiture, I think, and said, "Oh, look, you've got it inside out."
Watson's memory had let him down over the amount of water in her DNA data. The water content is vital to the structure, so their model was a complete disaster.
Rosalind was tickled pink. She was right. The building of a model of a crystal structure was a waste of time until you'd let diffraction speak for itself. And that was hard work.
I don't know, one might say, well, why not? It's an exploration to make a model. Make a model, and if you make a bit of a fool of yourself in the process, why worry? You might be lucky.
The real frustrating thing about science is how stupid people can be so much of the time. And so Francis and I were really stupid.
Worse than that, they had incurred the wrath of the London team's boss, Sir John Randall, who called up Watson and Crick's boss, Sir Lawrence Bragg, to complain about their behaviour. And Bragg was furious.
In those days, it wasn't gentlemanly to have knowledge of somebody's current unpublished work and to make use of that working on the same problem. It was rather like having an affair with his wife. It happened, but you didn't really take much credit in doing it.
Watson and Crick were kicked off the case, and even their model building equipment was sent to King's.
When we gave it up, we gave them the jigs for making models, and they didn't build any. So they never took on what we had failed to do.
Watson and Crick were officially out of the race. The way should have been open for the King's College team, but one thing stood in their path. Morris and Rosalind didn't get on. Why couldn't these two characters resolve their differences?
Morris was so shy that when he was talking to you and he didn't know you, he habitually talked at an angle. So you might find that you were addressing the back of his head.
I think scientists in particular tend to be rather, well, I don't know, sort of bottled up with serious thoughts and wonderful theories that, and the secret of life or something.
He would slide into a room and mumble something and be very diffident about it. He was never going to come in and say, well, I'm glad you've joined my team, and say, this is the way we do it, spit, spot, bang, which, had he done, would have cleared the air.
Oh, yeah, a number of these.
You know, he wasn't able to talk to her.
I think there are a number of people.
Rosalind was extraordinarily intelligent. Now, for that reason, she frightened the average bloke, I'm sure.
Today, Rosalind Franklin is an icon. Outside a building for female students named after her, there's a statue of her. To get a better sense of what she was like, we asked her sister, who apparently sounds similar to Rosalind, to read a letter she wrote to their religious father. She argues for the importance of science in understanding the world.
Science, for me, gives a partial explanation of life. Insofar as it goes, it is based on fact, experience, and experiment. Your theories are those which you and many other people find easiest and pleasantest to believe. But as far as I can see, they have no foundation other than that they lead to a pleasant view of life and an exaggerated view of our own importance. Anyone able to believe in all that religion implies obviously must have such faith. But I maintain that faith in this world is perfectly possible without faith in another world.
The picture, to me, seemed to say something about Rosalind. Rosalind has sometimes seemed a bit sort of heavy in appearance. I mean, not always, but sometimes. And I thought, well, it would have been nice if she'd been able to sort of trip around on her toes and look pretty and cheerful. But I think it was very sad because we had thought we might be all able to join together, you see, in the scientific work. There are two sides to the whole thing.
Back in Cambridge, Watson was down in the dumps, banned from working on DNA. His dream of showing it was the secret of life was slipping away. But Crick had some good news. Someone was coming to dinner in Cambridge who could help them.
The rest of us your favorite.
Erwin Shargaff had never wanted to help Watson and Crick. Even at 96 years old, he was still bitter about what had happened. Shargaff was an expert in the chemistry of DNA. Over dinner, Watson and Crick tried to plumb him for information.
They were fishing, really. But that, I get the impression, they did all the time. I think Watson really was a fisherman. I mean, he sort of brought the news to Crick. Crick was apparently the man who had the ideas.
Well, I think we didn't like him because he sort of didn't, warm up to our saying, you could solve the structure of DNA by model building,
They both showed an extreme contempt for chemistry. That was what struck me. They were more like children in their behavior.
He was just a born enemy.
Despite his extreme dislike for them, he did explain his Chargaff rules that state the relative amounts of the 4 basic ingredients of DNA. By comparing samples from 3 different species, he discovered a strange correlation. No matter what the life form, the amount of A equaled the amount of T, and the amount of C equaled the amount of G. This suggested the chemicals somehow went together in pairs. For Chargaff, this was an interesting correlation, but for Watson and Crick, it was the first clue to the structure.
I think DNA to him was an objective whose time had not yet come.
Here on this table here. Oh yes, good, yes. There's a unicam. Excellent.
On this desk is the original equipment that helped reveal the structure of DNA.
So this is the first camera, this is the first DNA.
It's worth a fortune today.
I mean, putting them in a frame like that, they look much better than just sort of lying on a table, don't they? The X-rays go in here, photographic film is put on the inside here, then that is put down around the DNA.
Like everything with Morris, there's more to it than meets the eye.
Now, you want to say something about these? These are small-scale models.
Morris had also wanted to build models based on the X-ray data, but Rosalind had all the best DNA samples, and they weren't talking. No one else at King's really had the imagination to help him.
At King's, if anybody did perhaps see the overarching picture, Wilkins did, because he often said, we should all wake up, we should all try a number of different things instead of plodding along trying to solve the X-ray diffraction pattern, because we are in a race. It is an important problem. And there are other people. I mean, there was this bogeyman sitting in America called Linus Pauling, who already had 2 Nobel Prizes, not 1, and he was thinking about it.
By now, Linus Pauling had turned his full attention to DNA. Suspiciously, this coincided with a certain young playboy travelling all the way from America to work in Watson and Crick's office.
In those days, Linus had a son who was about the same age as Watson, and knew Watson. And Pauling came across, came to our lab, and he went and stayed with Watson and Crick. And I think he put the fear of God into them, that Dad was thinking about this.
I had to feel, what was he doing in the laboratory of his father's big competitor?
I was accused of being a double agent, but I don't accept that, because I'd write to Pa and just say what I'm doing, and he'd write back, what he was interested in.
And Pa was not only writing back to Peter, he was busy writing other letters too.
I wrote to Wilkins at King's College asking if I could have prints of the photographs that he had obtained, but my effort was not successful.
By now, King's College had this X pattern, which indicates that DNA is a spiral or helix shape. It was Rosalind Franklin's best photo yet, but she wasn't letting anyone else see it. So Pauling never saw the X.
Could he have come up with the right answer without seeing the King's data? And of course, I hoped he couldn't. It wasn't until, about the last day of January that Peter came in after lunch and had a manuscript.
I had a letter from my father in December '52 saying he had proposed a structure for DNA. So I told the boys, they rushed over.
My stomach sank, and I was scared to death. So what was going to be in it?
Opened it and read it, and they discovered, that it was wrong.
I wonder what got into Linus.
Pauling's model not only didn't fit the data, it also failed to explain anything about what DNA did. He had blundered by trying to get the structure with too little information.
There wasn't any ambiguity as to whether Linus was right or wrong. He said Linus is wrong. So we were both pleased and a bit scared because maybe someone at Caltech Script would tell Linus this is chemical nonsense. Little did we know that, no one at Caltech really had the courage to tell Linus he was wrong. Linus was like the Pope. Linus wasn't used to people saying he was wrong.
So I've had a very fortunate life.
I thought the people at King's should be relieved. So without being asked, I thought, well, I'll just take the manuscript down to King's. I was just bubbling over with the fact that we had another chance. Pauling had got it wrong, and we should go into action fast. So I went down and looked for Morris and didn't spot him, and someone told me where Rosalind's office was. So I went toward it and walked in. She wasn't there. I wasn't trying to read the letters on her desk or anything like that, but obviously I was looking around, and she came in. Someone had told her that I was looking for her. She had a very negative reaction to me, and fury was rising. She didn't think I should be there. So I got out of the room as fast as possible, and then Morris Wilkins had heard I was around, and there he was, and I said, oh, I thought she was going to hit me, and he said, oh, I thought she was going to do that to me once. And so I went to, he took me to his office and opened a drawer and took out a photo, and there it was, the cross, which I had never seen and which they had basically weren't talking about.
It showed this sort of X type of OXO type of cross pattern, which was an indication of a helix.
I was on a big high. I mean, it was the most beautiful photograph. It's as if you've seen the most beautiful girl in the world and you're going to see her again. It was great.
If I got excited about the results, I tended to pass them on. I don't know, I think I didn't feel there was any sort of bombshell in this.
Well, the picture kept sort of racing through my brain, and I wanted to be sure I had it right, so I wrote it down. It was just a super reflection at 3.4 angstroms, and boom, boom, boom. No one could look at it and say it's not a helix.
And the reason that Watson realized that it was a helix?
So as you get a cross in the diffraction pattern.
Well, it just so happened that Crick knew this bit of X-ray diffraction theory and had told Watson that an X indicated a helix. Crick's mind was able to absorb bits and pieces from all over the place, and now this was paying off for them.
I felt, maybe we'll get the answer. Until then, I didn't feel we were close. I felt that picture means we're close.
Wilkins undoubtedly, and I think if you ask him, he will say that he feels that he did. If there were any cats to be let out of any bags, he'd done it.
Well, that's perfectly true, but I think this science isn't supposed to be kept in bags, no more than cats. I mean, I don't know what he means, but I don't like it as a scientist to sort of working away and sort of saying, oh, no, I mustn't tell the other scientists or something. I don't think it's the way to be working. Science ought to be an open activity, so you can work as a community.
It's been impossible not to build bottles then after seeing that picture.
Spring came early to Cambridge in 1953. Watson and Crick had been given official permission to return to their work on DNA, and by now they'd managed to acquire all the information they needed. Franklin's photo suggested DNA was some kind of helix. Chargaff's data suggested the 4 chemicals in DNA might go together in pairs. But what was the structure? What was DNA?
The Cavendish shop was to build us some tin models, and that took too long. And, finally in desperation, I made some out of cardboard. I sort of finished the job on Friday and didn't get back into the Cavendish probably until much before 9:30 on Saturday morning. So I came in the morning and I began moving them around. And I wanted an arrangement, where I had a big and a small. So how did you do it? Somehow you had to form link bonds. So here's A and here's T. And I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Ooh. So then I went to the pair, and I wanted this nitrogen to point to this one. I went like this. Boom. They look the same. And you can put one right on top of the other.
The two together make the same shape and can go on top of each other like the steps in a staircase, or to form a helix, a spiral staircase.
So you could have a small one, a big one, a small one, any sequence. If you knew the sequence of one, you knew the sequence of the other. We knew we could just, even if we go up to the ceiling, we were building a tiny fraction of a molecule. 100 million of these base pairs in one molecule. So unbelievably, all united by this either AT or TA, GC, CG, all fitting into this wonderful symmetry which we saw the morning of February 28th, 1953.
What they had was a double helix, a structure that revealed far more about the way life works than they could ever have dreamed of. They'd been looking for something that could divide, just like cells do, and it was easy to see how a double helix could unwind and form 2 more double helixes. They've been looking to see if DNA was the script or instructions for all living things. They realized that the millions of G's, A's, T's, and C's must be written in some kind of code, the script of life. And they even saw how the script could be copied exactly. As the double helix unwinds, each of the letters forms a new pair. And because A always goes with T and G always goes with C, the resulting 2 pieces of DNA are exact copies of the original, enabling the script to be passed from cell to cell and ultimately from generation to generation. It was clear now, DNA was the molecule that controlled all living things. Watson and Crick ran straight to the pub, where the locals were going to find their news hard to believe.
It was hard to contain the fact that, maybe we had a gigantic breakthrough. We'd done something really important. We had discovered the secret of life. You could say that day was the beginning of the new genetics.
They had the idea, but now they wanted to check that it was right. They set about building a tin model as quickly as they could, cross-checking their coordinates with the King's data. It all fitted.
Then we had to tell the people at King's, and we were a bit apprehensive because we didn't want to say, well, we've beaten you.
You have to remember that I'd been up before and seen the model that was wrong. And And that gave us a buzz and a high. And then you go up there and you see this thing. It looked right when you saw it. It was so brilliantly, elegantly simple. I thought, oh my God, we got scooped. Because I really thought we were going to come up with something like that ourselves.
That was terrible, you know, poor Morris.
I said that the double helix was somewhat similar to a young baby standing there, all alive, and saying, I don't care what you say or what you think. I know I am right.
Rosalind would have been appalled to learn that they had taken quite so much detail of her current work and put it into their model.
I thought, you know, we behaved as sort of the average person would behave. And we just thought, the people at King's haven't done the obvious thing. Why didn't they try using their data with asking what the molecule wanted to do?
Rosalind Franklin kept her distance from the other characters in this story. She spent her time working alone in her lab. People have wondered how close she got to finding the structure of DNA. The only way to know is to visit this archive in Cambridge, where her notebooks are kept. Her notes suggest that she nearly got there. In one of her final entries, she is thinking in terms of a two-chain or double helix. But that's as far as she got. She died just 5 years later without ever being told the extent to which Watson and Crick used her data. And when the Nobel Prizes were awarded for the discovery, she didn't get one, because Nobel Prizes aren't awarded posthumously. People that knew her say that what she cared about most was that her work moved science forward. It was one of the few things she had in common with Maurice Wilkins. Without their work, Watson and Crick could not have built this model. And the model was just the beginning. Over the next 2 decades, scientists delved into the molecular world of DNA and discovered how it actually controls life. But the person who did more than anyone to unravel its mysteries was a genius called Francis Crick. Today he's at the Salk Institute, California. In one interview, he did talk about what for him came after the double helix. It involves thinking beyond what he calls the narrow limits of normal human experience.
Now, when you want to understand the world, you have to go beyond those narrow limits, both in up and down, both in space and time. And then you find that there's a uniformity and extraordinary things happening which you'd no idea of just looking at the world. And this is the fascination of science, really, I think, to uncover so much which is not apparent just in everyday life.
Today, we can see the molecular world. This is a computer representation of DNA, based on accurate data. It's a long way from ball and spoke model building. What was apparent was that everything in the molecular world is more strange and sophisticated than anyone had thought.
Biological systems are the result of evolution, and they produce very complicated things. Now, the reason that DNA looks so beautiful and simple is it goes right back to near the origins of life, where things had to be simple. But if you actually look at the actual process of DNA replication, it isn't at all the way that we used to describe it. All sorts of funny things happen. You have to have proteins which will unwind the helix and nick it and then join it together again. You get an enormously baroque, complicated apparatus, which one might say molecular gadgetry, which actually does the job.
This is how the chemical code of DNA copies itself. But Crick was focusing on what the DNA code ultimately did, and that involved unpicking the gadgets of the molecular world.
What's sometimes called reverse engineering. It happens in the commercial world when one firm produces a gadget and another firm buys it and tries to take it to pieces and find how it works. That's called reverse engineering. But in our case, it's reverse engineering what you might call a foreign culture.
As a result of that process, it's possible to see how DNA makes living things, how a simple code is turned into flesh and blood. The blue molecule racing down the DNA unzips the double helix and copies one of the two strands. This copy is then released for the next stage of the process. The yellow copy feeds into another machine which deciphers the code and orders up the right components, one unit at a time, from the surrounding chemical soup. The product of this machine is protein. This could be 1,000th of an eyebrow. The same process could make a tiger's claw or part of the wings of a dove. It's the same for all life. What is made is all down to the DNA, the specific order of the chemical code. And today these machines can read the DNA code. How a person is made is being revealed. How a brain is built is being explained. In the G's, A's, T's and C's, they are beginning to read differences in our characters and personality.
The genetics of human nature is slowly unfolding.
Even our story can be seen in this way. The fate of our characters determined by their individual natures.
I thought the problem would last me my lifetime. I had no idea it would be solved within 20 years. You see, it was embarrassing almost to got to the stage where instead of this problem lasting for one's life, one had to look around for another problem.
So now Francis Crick is trying to find out how the brain works. He has always just been interested in making new discoveries. Jim Watson is president of Cold Spring Harbor Laboratories. He's built a house on the grounds with a statue of the double helix in his garden. He gives lectures, arguing for the benefits of genetic engineering.
There are people who say, well, we're playing God. And, I have a straightforward answer. If we don't play God, who will?
And Maurice Wilkins couldn't be more different. Having worked on the atomic bomb, he chose to study DNA because he thought it wouldn't be controversial. Today, as the newspapers talk of creating designer babies and the birth of human clones, he lectures at King's College London about the social responsibility of science. He hopes the human race will use this knowledge wisely.